Hello, good afternoon, everyone who all are joining in and who may join later in the session. This is your host, Himanshi Kaushik, and I sincerely and warmly welcome you all in today's session on technological enhancement for nutrient supply in cannabis plant species. And before we move on to the session and learn a lot more about the basically cannabis species and the nutrient supply and how we can enhance the species, we are joined by our experts here. And before I request Gurmeen ma'am to introduce us to our expert here, I hope that you all are doing fine, safe, sound, and happy. Now, I request Gurmeen ma'am to kindly please introduce our expert to us.
Very good afternoon to all of you. In this ongoing series of webinars, today we have Dr. Anand Mohan, who is currently working as an associate professor in the School of Bioengineering and Biosciences. He did his PhD in environmental biotechnology and toxicology from Jamia Hamdard, New Delhi. His master's was from Department of Biotechnology, IIT Roorkee. He is having a total of 12 years of teaching experience at university level for teaching undergraduate and postgraduate classes. He is having more than 20 years of research experience and is having a total of 40 research papers and chapters to his credit. He has qualified CSIR, UGC NET, JRF 3 times, and he has guided 5 PhD candidates, and currently 7 students are working under his supervision. He was awarded with the Research Appreciation Award by LPU in 2018. He also won a 3rd prize in the oral presentation on National Conference on Bioprospecting organized by the AMI unit at LPU. So now I would request Dr. Anand Mohan sir to please speak upon the topic technological enhancements for the nutrient supply in cannabis plant species. Over to you, sir.
Okay, thank you, Gurmeen madam. Thank you for your nice introduction. Students, as we can see here, there are specific enhancement going on right now. Okay, there is a high level of technological enhancement going for the nutrient supply in different plant species. So currently in this topic, I am going to talk about how the nutrient supply could be increased in cannabis species, what are the different type of aspects for the nutrient degradation in different plants. So as we can see here, why is mineral nutrition important? So in most of the natural soils, as we can see that the availability of mineral nutrients are very much responsible for the plant growth and its productivity. Nutrient limitation is an important selective pressure, and the plant may face challenges regarding how to acquire and use the mineral nutrients efficiently. Plant nutrition specifically does not refer to photosynthesis because this is something different than the photosynthesis. Now, I would give a brief introduction. What are the different type of elements? So basically, essential elements are there which are required by the plants to carry out their natural life cycle. So in the absence of these essential elements, plants cannot complete a normal life cycle. There are specific elements which can be defined here. So there you can see here there are 17 elements which are classified as macronutrients and micronutrients. The macronutrients are those nutrients which would be present at a concentration more than 10 millimolar per kg dry weight, while the micronutrients should be present at below these concentrations. So if we do a hydroponic experiment, we can see here that mineral nutrients are required, essential nutrients, and as we can see here in the current figure, the complete solution containing all minerals ultimately will promote a good and healthy plant, while a solution lacking potassium would produce a plant having chlorosis. Chlorosis means a yellowish spot on the plant. Now, what are the macronutrients? So macronutrients are there total 9 in number, and these are carbon, oxygen, hydrogen, nitrogen, sulfur, phosphorus, etc. The other 3, potassium, calcium, and magnesium, are also required. Further, micronutrients, what are these micronutrients? The micronutrients are required in lesser amounts. Now, there are 8 number of total micronutrients: the iron, chlorine, copper, zinc, manganese, molybdenum, boron, nickel, etc. Now, these micronutrients basically act as cofactors for the enzymatic reactions. Basically, there are different kind of metabolic reactions occurring inside the plant body. Now, these metabolic reactions require a large number of enzymes, and to help these enzymes achieve their function, the cofactors are required. So these micronutrients basically they act as cofactors. Now, as we can see here in this table, we can see there are primary macronutrients, secondary macronutrients, and micronutrients. So now what are primary macronutrients? The primary macronutrients basically are those nutrients which are required in much larger amount, which are required in the production of different structural molecules inside the plant body. Like for example, nitrogen. Nitrogen is represented by N and it is utilized to produce the proteins, amino acids, etc. While the next important compound is the phosphorus, which is required to produce the nucleic acids and adenosine triphosphate, that is the energy currency of the cell. Further, the next important compound is potassium, which is represented as K. It is utilized as a catalyst in ion transportation methods. Further, there are secondary macronutrients which are required in lesser amounts, as we can see here. Calcium, which is represented as Ca, and it is an important component of cell wall. Magnesium, it is the part of chlorophyll. Sulfur, it is the part of amino acids. The iron, which is required for the chlorophyll synthesis. So as we can see here, we have discussed right now till the primary macronutrients and the secondary macronutrients. So they would be required in larger amounts. They would be required in the structural composition of different kinds of plant parts. The further micronutrients are there. So these micronutrients include copper, manganese, zinc, boron, molybdenum, chlorine, etc. So as discussed earlier as well, these micronutrients are basically, they could be component of enzymes, or they could be activating the enzymes, or they could be cell wall components. Molybdenum, as we can see here, it is involved in the nitrogen fixation reaction. Now, as we can see here, if the micronutrients are present in lesser amounts, ultimately some of the deficiency symptoms could be shown inside the plants. Okay, so these deficiency symptoms would be in the form of yellowing of the leaves. It would be in the form of yellowing of the leaves. Like for example, if we see nitrogen, now deficiency symptom would be in the form of chlorosis of the leaves. That is, the leaves will become yellow and they will further die. So as we can see here, the figure associated here, we can see the leaves have turned yellow and then further they will die. The younger leaves will remain green because they would be receiving the new nitrogen supply from the older leaves. So this would be the deficiency symptom for nitrogen. Similarly, the deficiency symptom for phosphorus, again, we can see here, the phosphorus deficient plants are stunted, they stop, the growth is stopped, and ultimately the phosphorus is also redistributed. So they show stunted growth, deficiency symptoms, etc. Further, we can see sulfur. Now in the sulfur, again, the chlorosis of leaves occurs. One of the important aspects of the chlorosis of leaves in the case of the sulfur deficiency is that the vascular bundles also get chlorosis. Okay. Further, sulfur is not easily redistributed from the mature tissue, so its deficiency is usually noted firstly in younger leaves. So the young leaves will become yellow first. Now potassium deficiency is shown here. The leaves develop necrotic lesions and light chlorosis. The necrotic lesions will cause the necrosis of the tissue of the leaves and degradation of the tissue. Further, yellowing will occur and that would be called as the chlorosis. The tips, one of the important aspects of the deficiency symptom in the potassium is that the tips would die first, they would be showing the necrotic symptoms first. In the case of the calcium, the deficiency symptoms are represented as the meristematic regions die. Now, what are the meristematic regions in the plants? So as we can see here, the meristematic regions are those regions which are the growing part of the plant. They are growing fastly. So we can see here margins of the younger leaves become chlorotic, then necrotic. So margins, the outer margins of the leaves could be becoming chlorotic. They would be becoming yellow. The young leaves would be distorted and they would not be, sorry, they would be showing the distortion. In the case of the magnesium, the deficiency causes extensive intervenal chlorosis, which starts with basal leaves, that is the older leaves, and progresses to younger leaves. So we can see here the intervenal chlorosis means in between the veins, the chlorosis would be observed. Now the next one is the iron deficiency. As we can see here, in the case of the iron deficiency, again intervenal chlorosis would be observed, and as we can see here in the figure, the veins, the area in between the veins would become yellow. In the case of the copper, the deficiency, we can see they are rarely deficient and they become distorted and dark green in color. Now further, to make up with the aspect of the proper nutrition of the plant, we are doing important research in the university. Now we are working on some of the important nanofertilizer aspects. Now nanofertilizer for the fertilizers, which are basically synthesized in the nanoforms, in the nano molecular level, and further they could be utilized very judiciously. So now as we can see here, one of the important fertilizers we have focused on that are the nitrogen and phosphorus. So as we all knew that the plant requirement in the form of the nitrogen is fulfilled by utilizing the chemical fertilizer that is called as the urea. Now, in the case of the utilization of the urea, what happens is that a large amount of the urea is lost in the soil and ultimately very less is made available to the plant. So as we can see here, continuous years, ultimately the more and more urea is required and less urea is produced by our country. Now what happens is that the extensive use of these chemical fertilizers leads to irreversible harm to the soil structure. The use of synthetic nitrogen fertilizer in agriculture causes release of the greenhouse gases. Now as we know, the greenhouse gases and the environmental concerns are right now top important environmental concerns for the whole world. So as shown here in this figure, if we spray 100% fertilizer, out of this 100% fertilizer, the 70% is lost inside the soil, only 30% is utilized by the plant. So the 70% lost fertilizer in the form of surface water loss, it goes into the rivers, it goes into the lakes, it leaches down into the soil, it joins the groundwater. The 30% fertilizer is only utilized by the plants through other inorganic compounds. Now, our model plant, we have taken a model plant that is called as the Cannabis sativa. Now, Cannabis sativa is a very important plant growing as a weed, and also it is growing in the fields also. It produces a very important textile fiber that is called as the hemp. This Cannabis sativa is also utilized for the production of the narcotic substances which are utilized in the regenerative therapies, etc. Further, they also produce many of the important compounds which could be utilized in different medicinal aspects. Now, this Cannabis sativa plant could be characterized by long, narrow leaves with serrated leaf shape, tall and thin structure. So we can see here, this is the cannabis representative cannabis plant. Now, the cannabis plant is a valuable biomass crop in textile industry due to its high textile tensile strength fibers, as I have told you already. Production of the hemp. The government of India is also working on the production of hemp for extensive utilization and for the concentration of the economy. Now, one of the strategies we have utilized here for providing both the phosphorus and the nitrogen fertilizer to the cannabis plant is that we have synthesized a nano fertilizer by mixing two compounds, that is the urea, that is the normal urea, along with hydroxyapatite. Now, this hydroxyapatite is a compound of calcium and phosphorus which mixes together. So we have utilized a specific strategy for producing this UHAPF. So this is after synthesis, this would be called as the nano UHAPF. Now, when this UHAPF fertilizer is provided in the soil, it breaks down simultaneously, provides nitrogen to the plant, which is provided continuously for a longer period of time, such as almost 40 days. Okay, so the major difference comes here is that if we are utilizing a commercial urea, that commercial urea would be completely available to the plant within 2 or 3 days, and very less of that amount would be utilized, would be absorbed by the plant. While as seen before in the slide, almost 70% would be lost. Now here in our strategy, ultimately, since the urea is released slowly for a longer period of time, up to 40 days, what happens is that all of that is absorbed by the plant. Now, along with the nitrogen, the phosphorus available in the hydroxyapatite is also utilized by the utilization of different biofertilizer based microbes, like the Bacillus megaterium and Pseudomonas aeruginosa. So as we can see here in this figure, both the hydroxyapatite based nanofertilizer is provided to the plant along with these microbes, important microbes, so that at the same time with a single compound, we are providing both nitrogen and phosphorus to the plant. Now you can see here, this is a figure where the hydroxyapatite was synthesized. This is the nanostructure. As we can see the same images of the nanosynthesized urea hydroxyapatite fertilizer. These are the different bacteria we have used in our work, the Bacillus and the Pseudomonas. Now these Bacillus and Pseudomonas help in mobilizing the phosphorus from the hydroxyapatite fertilizer. Now, as we can see here, we have grown this cannabis plant in different sets, and as we can see here, the different samples were grouped in the form of the first one is the control, the second one is the urea only, the third one is the synthesized urea hydroxyapatite nanofertilizer, the fourth one is the Bacillus megaterium, the fifth one is the Pseudomonas aeruginosa, the sixth one is both microbe Bacillus megaterium and the nanofertilizer. The sixth one is the Pseudomonas aeruginosa, that is the bacteria 2, and the hydroxyapatite fertilizer. So as we can see here, this is a series of steps. These are the series of treatment groups that we have utilized here to enhance the nutrient supply to this plant. Now the major outcome from this study was that the urea hydroxyapatite fertilizer and our study groups provided higher amount of nutrition to the plant. They reduced the nutrient losses and synchronized the nutrient availability with the uptake requirement of the crops. The slow release functionalized urea hydroxyapatite fertilizer demonstrates that nitrogen release could be going on for more than 30 days period of time as compared to the commercially available urea. While the synergistic effect of the urea hydroxyapatite fertilizer and the microbial consortia would enhance the overall development of the hemp crop. So we can see here the hemp crop or the cannabis crop greatly benefits from the urea hydroxyapatite fertilizer treatment and delivers a slow and sustained release of essential nutrients, improving plant growth, biomass production, nutrient uptake efficiency, and no loss, no harmful input into the environment. So this was my presentation regarding the nutrient supply to the plant. Now further, I have some of the samples showing you the different plants which are having some or the other deficiency in the case. Okay, so right to the next figure. Yes. So as you can see here, the first figure we are showing here, the tip of this leaf is degraded, and ultimately it is showing up the signs of yellowing. Now this sample represents a perfect example of potassium based deficiency in the plant. So this plant, the leaf of which this plant is there, so ultimately this leaf was having potassium deficiency. Now in the case of the next sample, again we can see the tip of this of the leaves of this plant, they are having again necrosis and again yellowing in between. This yellowing and the necrosis of the tip again represents the potassium deficiency. Now the next sample. Here in this sample, we can see that in between the vascularization, ultimately there is yellowing. The vascularization is still having green coloration, while in between the tissue has become yellowed, and it is showing the signs of chlorosis. Now this is a perfect example of the sample for the molybdenum. Now the next sample. Now here in this sample we can see that the structure of the leaves, they have got distorted. Okay, the leaves are green. None of the leaves have turned yellow. But the structure has become more and more distorted. They are not properly arranged. Now, this is a perfect example for the deficiency symptom of copper element. Now we move to the next sample. As here we can see in this sample, again the chlorosis has occurred in between the vascular bundles, the tissues in between the vascular bundles. So it represents the sulfur-based deficiency in the leaves. Now further, I will move and show you the hydroxyapatite nanofertilizer that we have synthesized. So this is the powder-based hydroxyapatite nanofertilizer which we have synthesized in our laboratory, and this will supply both nitrogen and phosphorus to the cannabis plant at the same time. So we can see here, this is an important technological advancement in the field of the fertilizer production, especially in the field of the nano fertilizer production. Thank you very much to the audience for listening to my seminar.
Thank you, sir. Thank you so much. First of all, for giving that presentation. It was very informative presentation for me also. And after that, showing a practical demonstration as to what all the diseases, if I may call them, looks like in plants. And so there are some questions that I just want to take up. So, looking at the plants and the demonstration that we have shown here, it is, I think, in every plant we can see all these issues, right? Like any plant can have nitrogen deficiency or any other thing deficiency. So are there any ways where we can actually cure the plants naturally instead of using different chemicals? We can, let's say, try to give more water, maybe, create an, let's say, an environment so that plants do not have these deficiency.
Basically, as we can see that if we are growing the plant, suppose we are growing the plant in the field or the garden, we can try to give the plants a balanced nutrition. We can utilize organic components. The peat is there, one of the important organic component. Peat is available in the market. It provides all the essential nutrients. Further, we can utilize organic manure, which will also provide all the important components so that the plants can grow healthy and without any chlorosis or any of the deficiency symptoms.
Okay. And sir, how like in percentage, if I talk about how much we can cure them, as in, see, we know that environment is readily getting degraded due to whatever reasons, because of the, let's say, global warming, and now it is been released that after global warming we are in the critical phase now. So is there any way where we can create an artificial environment for the plants that is somehow a natural environment also? I mean, it is a contradictory thing, but again, I mean, we can give a little environmental friendly things to the plant. For example, we can control sunlight, or let's say water, or the amount of nutrients we're giving. So can we control all these things?
Definitely, we can do this, all of these things in a greenhouse. So
Okay.
in a greenhouse already, if we see that in the agricultural laboratory, universities, etc., even our lovely professional university is having a very large greenhouse facility. So where we can grow the plants in a controlled way. They would be provided with the proper amount of sunlight. They would be provided with the proper amount of carbon dioxide, along with the proper amount of different type of nutrients, so that we can grow up healthy plants with the healthy products.
Right, sir. Thank you. And with this, I just want all attendees to know that if you are concerned about plants in an environment, let's say, and if you want to, like people save humans, you want to save plants, then you can come in here. If not join, you can just take a look around and see how advanced we have been in technology, in terms of technology, or in terms of, giving, let's say, technological things to the plants as well, and where you can actually learn a lot about plants, a lot about the subject, and you will get a lot of guidance from our experts here. Anand sir is here, and we are having different experts for specific fields, and you can just take all the things and can excel in the field that you want to. And so with this, we're done with the queries as well. Now I shall take your permission to wrap this session up.
Yeah, thank you very much.
Thank you,
Nancy.
sir, for joining in, for crediting your time for all of us. And thank you, Sabri, who was helping us to demonstrate all the practical to all the students here. And all at the end, I also want to thank attendees for coming in here, and the questions you have given, or the interactions if you have given is really, really appreciated. With this, I take your leave, and maybe we meet soon next time in the in the university physically, and if not, then we'll definitely meet in the next webinar. Until then, stay safe, stay healthy, and thank you so much for joining in. Thank you so much.